I'm going to be talking about optical sensing, and we're going to be talking about optical sensing on a benchtop scale, spatial scale. But before we get into that, I just want to make a couple of important claims. First of all, I want to argue that I think optical sensing, the use of imaging technology, depending on the areas, it's called machine vision, remote sensing. It's all based on the same thing. We use different sensor technology to remotely characterize different objects at different spatial scales. And I will argue that is becoming the new molecular area. Just like in the 90s, the availability of molecular tools started completely changing how we are performing research. I think imaging technology is doing the same thing. So I want to make that claim. And the second claim I want to make with this is with the visuals here is that we are essentially, from the imaging technology, from the sensors we're using, we are acquiring very high value, high spectral resolution imaging data. So we get these reflectance profiles that can inform us about objects in different ways. And this is not black magic. There is a lot of physics. There's a lot of math behind this. So even though it may in some ways sound and appear to be somewhat what's going on here, it's a little bit like the initial in the first years of the molecular technology. I think we are still working out some of the details, but there's some fundamental physics and phenomena behind it, which I will talk briefly about, to hopefully convince you that this is not black magic, that this is worthwhile, and there's a reason why the military, pharmaceutical industry, the medical area, the mining industries, all these, and the agricultural sectors, of course, all are embracing imaging technologies at different spatial scales, using this data in all sorts of ways. And this is really an opportunity for us as entomologists to jump on that wagon and start integrating it into what we do and how we inspire our students. Optical sensing is the use of imaging technology, and typically we can mount that imaging technology on different, either in a stationary way, on a benchtop scale, and that's what I'm going to be talking about today. But fundamentally, it's the same technology we use when we deploy it with a drone, with a rover, with a satellite. We have many options at our disposal here, and fundamentally, it's the same thing. There are, of course, very important differences in addition to the spatial scale, and one of them being that if we are outside and we are dependent on solar light, then that the composition and the intensity of sunlight changes and the atmosphere changes and all that. So there's some calibration that needs to be included into this. But fundamentally, the use of the technology is the same at all these spatial scales. But I'm going to be talking about it specifically when we're in the lab and we have a camera that is mounted to acquire data at high spatial resolution, so from insects themselves, and at high spectral resolution where we have lots of spectral bands associated with each pixel. Probably some of the main reasons why optical sensing is being adopted more and more widely is because this technology allows for what's called high throughput. It's very fast. You can process and acquire a lot of data in a short amount of time. It's therefore cost effective. You can replace mechanisms and procedures that are typically very subjective and based on visual inspection or others. You can automate it, you can make it repeatable, you can also make it quantitative. And then lastly, and that's really important, it is non-destructive, meaning we can look at objects without damaging them. We don't have to take tissue samples and crush them up and then therefore lose them. We can actually just have a sensor, look at them, examine them by collecting their reflectance or transmission data, and then use that data to make some characterization of that particular object. So it's used very widely in quality control, classification, sorting, and again at multiple spatial scales, right? So if this is part of conservation efforts to find out what is the health status of a vegetation where we have insect communities that we are trying to preserve, and as I will talk about here, we can also look at the insects themselves and actually take images of them and use the reflectance to say something about them, and they can be both dead and alive. A key aspect in applications of optical sensing technologies is that we are often looking for the outliers. We are looking for that needle in a haystack. We are either scouting through or processing through a sample of insects, and we're trying to find that unique insect, that capra beetle, or that insect that is unique to us. Or we are flying over a crop field and we are looking for that hot spot where the spider mites seem to have established in a strawberry field. So it's the combination of fast, automated, repeatable, quantitative, all those things allows us to, in a fast way, process and find the outliers that we are typically looking for. That's what this technology really lends itself well to. An aspect of optical sensing that is extremely important for the use in entomological research and teaching is that even though we think of we acquire surface reflectance, we point the imaging device onto the surface of something, just like in this case the hand we project the infrared thermometer onto the hand. What we're really doing is we are assessing what's going on inside the hand with that thermometer. So what is important is that surface reflectance data also actually includes information about the tissue layers underneath the surface. So over on this curve here, or on this figure, you can see that it's particularly bands in the 800 and 900 nanometer range, so out in the near infrared, will actually penetrate several millimeters into lung tissue. So this is widely used in the medical field in their research. We as entomologists can use this because it means that we can, when we have sensors that acquire data in those wavelengths, we can actually quote unquote look into the insects. It's not like X-ray where we can actually outline the individual organs and whatnot, but we can get some information from the internal status of the insect, and that is critically important when we want to look at differences, say, between males and females, even if the morphological features are very, very difficult to distinguish. Maybe they have different fat body content, maybe they vary in some other ways that can be big or small, but at least it explains why it's possible, based on optical sensing data, to make inferences about these objects and how they vary. As an example of a proof of this penetration that we are looking inside objects, I'm going to tell a little story here. So I've already mentioned that I'm from Denmark and we like Lego. Another thing we like is Hans Christian Andersen and the fairy tales that he wrote. And one of them, of course, is about princesses and how you find out whether a woman is a princess. And so this is the story about the princess and the pea, right? So we all know now that if you want to know if somebody is a princess, you have to offer her to sleep somewhere and then put a pea underneath a pile of mattresses. And if she wakes up and is all sore in her back, well, there you go, you have a princess. So inspired by this story, I published this experiment, actually, where I had 3 colored dots. These are my peas here, a blue and a green and a red, printed on a piece of paper. And then I put mattresses on top. Those, in this particular case, it was just sheets of paper. And so the question I wanted to find out was, how many pieces of paper would I need to stack on top of my colored dots for the imaging data that I collected right on top of the dots, for those those data no longer to show effect of the dots, of the P's, so to speak. And the graph to the right actually shows the results. So if the P was blue, it only took me about 5 sheets of paper for that dot to disappear. But if it was green and even red, I would have as many as as many as 30 sheets of paper. And what you can see here is that the highest number of sheets of paper were needed in the 800 to 900 nanometers, which is exactly the data I was showing you prior on the other slide from lung tissue, where the medical field are seeing the same thing. So whether we're looking at lung tissue or whether we're looking at lignified paper, we're seeing the same thing, that the penetration of these spectral bands is highest in that 800 to 950 nanometers. And that just confirms that we can look into insects and learn something about their internal structure, their internal composition. And that is precisely the foundation for what we are trying to accomplish when we use optical sensing in classification of insects, or when we look at whether they, for instance, in this particular that I'll show here in a little bit, is whether they are vectors of plant pathogens. There's a couple of important take-home messages from this study that was inspired by Hans Christian Andersen. And first of all, so I demonstrated through a series of experimental studies where some of them included insects, but some of it actually didn't. But they were critically important for me to afterwards make the claim and continue to make the claim that we can, quote unquote, look inside insects. And the second claim I want to, or take home message I want to bring up here is that because I was using a technology and sort of a cutting edge technology, I had to venture outside the conventional entomology zone. I had to sort of accept that I needed to go outside the comfort zone of where I am as an entomologist. So that was clearly needed. And I think we are seeing that more and more across entomological disciplines, that people are now, because of what they do, have to go in different directions and venture out and work with people, that economists or with programmers or engineers or others, in ways that we haven't seen much of in the past, but we will see more and more of. And finally, this whole study was clearly driven by the technology. I didn't think of this initially as something I needed to do, but in order for me to use the technology properly after this, I needed to inform myself. I had to do the experiments, and then I decided to write a paper about penetration and scattering, about optical phenomena as an entomologist, right? So you can imagine how much deep water I was on here, but I got away with it. It took a while to get it published, but I did. But that's clearly, to me, an example of how the technology is now driving research rather than the other way around, the way it was in the past. A couple of years ago, we reviewed the technology of optical sensing, remote sensing, and its use in entomology. So in this review article from 2016, you can find lots of examples, lots of studies in which this technology has been used, species identification, for all sorts of physiological studies, and also in ways, for instance, to assess products with and without insects. So in the stored grain entomology area, with and without parasitism, they're just and symbiont biology. So lots of examples of how this technology has been applied and will be applied more and more in the future in entomology, in specific studies of insects, as well as on other spatial scales when we go out and look in agricultural areas and areas targeted for conservation and whatnot. In this study of beet leafhoppers, the context, of course, is that beet leafhoppers are the only known vectors of beet curly top virus to tomato and other crops in California. And growers are restricted in their ability to use insecticides to control beet leafhoppers. First of all, because lots of products are not labeled and many of them don't work particularly well. And thirdly, the biology of leafhoppers makes control of them very complicated because they live long and they migrate in over an extended period. So when exactly are you supposed to spray? Now, the leafhoppers, the beet leafhoppers themselves are not particularly damaging to the crop. It's when they carry the beet curly-top virus that is transmitted during feeding and probing. So just because you have leafhoppers in your tomato field doesn't necessarily mean that you have a problem. And that is really the big issue here. So could we come up with a technology that would allow us to sample leafhoppers and then in a fast, repeatable way have a diagnostic tool that could be used to determine, are we having a population that is carrying the beet curly-top virus that are virulliferous, or are these mainly non-virulliferous individuals? So it's also about not just presence, absence of the virus. It would be also very important if you sampled, say, 50 or 100 leafhoppers. Could you then find out, analyze them individually, and then find out what is the proportion of individuals? If you did the PCR analysis or some other sample, you would get a positive or a negative, but you wouldn't know how many of the individuals were actually carrying it. So if we could do this with imaging, there would be huge advantages to this. So that's why we started having some colonies of leafhoppers that we then established on different crops with and without the virus, and then we followed them over time. And what you can see to the right, and there are different aspects of this paper that I'm not going to talk about today, but what you can see is if we have positive controls, we could detect that with 100%. And we could then also, as the beet leafhoppers acquired it, as they acquired more and more, there was also a progressive increase in their infection rate, which is what we would expect. A couple of the values are a little bit off, and that is to be expected. That it is not nearly as accurate as PCR is. Of course it's not. But on the other hand, it's super fast, and it would be much cheaper in the long run, and you could get individual data. So instead of having to wait a long time and only have one data point from a group of individuals, you could then have individualized images from individual data points. So that gives you much higher resolution. Even though some of them may not be accurate, you're getting a much broader picture that at least could complement some PCR. Or you could argue that if you have a lot of samples, that maybe you could use it as a first type of screening. In other words, we're coming up with a technology here that could really help inform growers on when to spray, to avoid spraying when it's not necessary, and really use these pesticides when they absolutely have virulliferous populations threatening their specialty crop. What I think is really important to emphasize in this particular case is that we have a very important problem in front of us, a problem that growers and the tomato industry, the specialty crop industry is facing. And of course we do the research that then educates students and all that, and we get the study published. But along the way, we are clearly thinking beyond the publication, that the publication is one step, but the real goal is down the road to develop a diagnostic tool that can then be integrated into diagnostic labs or in other ways create job opportunities and professional training for our students. So it's very important in the way we approach this problem, not only to look at this, not simply just because we got a publication about this that we can then okay, check that box and now we're done. No, that's when the job actually really starts. Is now we have the credentials, now we have the knowledge base that allows us to go to really work with the problem, which is how do we take it to the next level? How do we develop something that growers can use? How can we get it out in the real world?